Hello, my name is Neo Ra for Physics 2212 of Spring 2026 semester, and this is my public lecture review for The Physics of Genes and the Promise of Personalized Medicine by Professor Alexey Aksimentiev. It was a lecture on physics concepts related to DNA molecules mostly, and it was presented on 2016, November 17th. Now we will delve into this lecture. Uh, well, this was interesting to me because I am a BME major, and in high school, I've learned a lot about DNA, RNA, and, well, mostly gene molecules in biological ways. However, the knowledge that I had of these molecules was limited since I was a high schooler and only studying biology. However, this lecture gave me some new things to learn about why these things are like that. So it was the most interesting lecture for me, and I selected this to be my public lecture review. For example, let's see. So, DNA is a highly negatively charged polymer that forms human genome. This was a concept that I've learned in high school biology already. However, this was the extent to which I learned, and I never learned why it's negatively charged or how it is negatively charged, because since DNA are all negatively charged despite being very closely packed, if they were both negatively charged, they would by effect repel each other, and therefore it would be problematic in DNA packing. In this lecture, Professor Aksimentiev explained it by multivalent, which has charge of plus 2e or more ions, serve as bridges connecting DNA. So here's a image that shows this very well. So for example, if we are just dealing with single valent ions that have plus 1 charge compared to DNA molecules that have minus 1 charge, then they would only be attracted to each other, and the compound, the DNA and the ion compound, would have no effect on each other. However, when we're dealing with multivalent ions like this, it has plus 2 charges, so it can hold on to both DNA molecules, and in effect, it's attracting these molecules together. So like this lecture provided me with lots of whys to things that I've already learned in high school. And then he goes on to explain the methylation, which is adding a methyl group to the gene molecules, which makes it have the same charge, however, but bigger size of the molecules that are being packed. And he also talks about DNA segments that are rich in A and T nucleotides, which also function as structural DNA instead of junk DNA that attracts and packs the DNA molecules together. And he then explains that these are both achieved by sharing polyamine. And then he explains a lot of technologies, and for these reasons, I selected this as my public lecture review. Thank you.